Today we're going to talk about the nerve-muscle relationship and the neuromuscular junction. Recall that skeletal muscle, which is a striated voluntary muscle, must be stimulated by a nerve, and specifically by a somatic motor neuron. The cell bodies of these are found in the brain and the spinal cord, right? And these axons of the somatic motor neurons extend all the way to the muscle cells. So you might have some neurons that are pretty darn long if it's talking to like my big toe. And each motor neuron can actually talk to multiple muscle cells, multiple muscle fibers it innervates, and that's called a motor unit. And the size of the motor unit, you can have maybe 5 muscle cells being talked to by a single motor neuron, or 500. And you can see that if the fewer number of motor cells, muscle cells that are talked to, the better the control is. You can get more distinct fine control versus gross motor movements. So for instance, the muscles of your face have relatively small motor units, one motor neuron talking to just a few cells, so you can just wiggle your eyes or bat your eyelashes or that little half smile. In contrast, muscles of the legs and the gluteus and the arms are going to be controlled by big motor units, by a single motor neuron talking to lots and lots and lots of muscle cells, because you need all the muscles to contract to take a step forward anyway. So that's kind of the situation where we're looking at the nerve-muscle relationship. Here you can see the end of an axon of a motor neuron talking to this motor neuron. And we're going to blow it up, and here you can see a single axon and a single synapse, and then the structure between them, the gap here, the synapse, and then the motor end plate. So we're going to look a little bit more closely at the important structures here. So the neuromuscular junction is a type of chemical synapse, and we often abbreviate it NMJ, neuromuscular junction. And it's simply where a neuron makes a connection with its target cell. And in this case, that target cell is the motor cell, the muscle cell. And you can see here, you can see a little bit of our myofibril, our sarcomere here, so that kind of tips you off to what type of target we're looking at. And here you have the motor neuron, and it's going to be surrounded by a Schwann cell. And you can see some Schwann cells here. And these are going to help insulate it and isolate it and keep the neurotransmitter from diffusing out of the synapse, as well as increase the speed of transmission of the information coming from the brain to the muscle cell itself. Now, these little blue dots are synaptic vesicles. All right, and you can see we have quite a few of them in here. And these synaptic vesicles store the neurotransmitter, the chemical that's going to be released from the motor neuron that tells the muscle to contract, that says, hey, we're going to stimulate you. And remember that muscle cells are excitable, and that means that they respond to stimulation with electrical activity. We're going to talk more about that in the next lesson. But here the stimulation is acetylcholine. All motor neurons release acetylcholine, which means what type of receptor does this muscle cell have to have? If the chemical is acetylcholine, what receptor do we want to have? That's right, we want to have acetylcholine receptors. So acetylcholine receptors are going to be found on the cell membrane of the muscle cell. So the components of the neuromuscular junction include the synaptic terminal. That's this structure here that contains the synaptic vesicles, that contains acetylcholine. And then we have acetylcholine receptors on the muscle cell. Now you'll notice that this cell membrane here of the muscle cell has these little pockets, these little invaginations, and that's going to increase surface area. So we're going to actually pack this full. All these little red lines are acetylcholine receptors. So you can see that the more receptors we have, the more likely it is that we're going to be able to respond to acetylcholine. The more likely it is that the muscle is going to respond and contract. We call this specialized area the motor end plate, sometimes called the MEP, the motor end plate. And you might see that in your reading. So the motor end plate is just the specialized location where the neuron talks to the muscle. Sometimes we just call that the synapse, right? This is extrasynaptic out here, and this is the synapse. So we have acetylcholine coming from the neuron. We have acetylcholine receptors on the muscle, and then we also have the acetylcholine esterase. Now, acetylcholine esterase is an enzyme, and it's extracellular, so it's actually out here in the synapse, and it degrades acetylcholine. Because as long as acetylcholine is present, then muscle cell is going to be excited, and that's going to stimulate contraction. So if I want to raise my hand, do I want to go ahead and leave my hand up for the rest of my life? No, right? I want to be able to bring it back down, to relax the muscles that enabled me to raise it. One method that we relax is we have to get rid of the acetylcholine. And one way we do that, and the primary way we do that, is we simply break it down. We break it down to component parts, and it can no longer activate the acetylcholine receptor. And the enzyme that does that is called acetylcholine esterase. So it degrades acetylcholine and helps and aids in relaxation. Now, some other terms that we need to talk about is just this idea of the synaptic cleft. That's just the gap between the nerve and the target muscles. The cells don't touch. They come very, very close, but they don't actually touch. So that's just what sometimes we just again abbreviate the synapse or the gap or this space, but it's sometimes called the synaptic cleft. And then we've already talked about Schwann cells. Schwann cells are glial cells that help envelop and isolate the neuromuscular junction and the neuron itself. And in the next slide, all I've done is blow this up. So here again, we see a Schwann cell. We have a couple of Schwann cells just shown here. You can see a little bit of a Schwann over here as well. We have lots of synaptic vessels that contain acetylcholine for release. Notice that they're right up against the membrane so that when we exocytose the synaptic vesicle, it's going to release its acetylcholine into the synaptic gap or the synaptic cleft. Now, if it's not going to bind to its receptors, we can show a few receptors here in our motor end plate. Then it's going to get degraded by acetylcholine esterase. And remember that this receptor, right, is what we call a ligand or chemically gated sodium channel. So that means that, if you remember from way back when, ligand gated or chemically gated, right, says that it opens in response to a chemical or to a ligand, all right, and then it lets in sodium. And remember that sodium's really high outside and really low inside most cells. And that's what we're going to talk about in the next unit and then the next lesson, to talk about how do we couple excitation, what's happening at the neuromuscular junction, with contraction, what's happening down here. And how do we make sure that those two things are always coupled? Because we don't want contraction to take place unless the motor neuron is telling us to, unless the brain is saying, we need you to contract for this reason. We don't want spontaneous contraction. That's called a cramp, and those are never fun. So in the next section, we'll go ahead and talk about excitation-contraction coupling, which is really the hallmark and really the nitty-gritty of how muscles do their job.